Okay, guys, we are finally going to leave the cell level and we're going to go up into the next level, which is tissues. And this is all chapter 5 kind of stuff. Now, you might wonder how it is that we know so much about what's in the cells. These are microscopic structures. Obviously, we need some kind of equipment to see these for real. And yet we know so much about the internal structures of a cell. Well, this all started with microscopy. It started with our ability to actually gain access visually to what was going on inside of these cells and to some of these subcellular structures like organelles. Okay, so this all came out of study of tissues. We were able to actually take tissues, which are collections of cells, of course, and we were able to image them through the study of tissues called histology. All right, so you might have seen in Blackboard under the Module 2 title and course content an image that looks kind of like this, and this is actually a histological slide. Okay, we use this to actually study the tissue. In this case, it was bone. Okay, this is actually a picture of a cross section of a bone. So histology is the study of tissues, basic definition, and one of the very rudimentary ways of going about this would be to use light microscopy. So some of the very earliest microscopes, even starting with Van Leeuwenhoek, who was a gentleman who actually had a really rudimentary microscope. He would use the light from his surroundings. He would use a very rudimentary magnifying lens, and he was able to look at structures. And I think he was able to actually look at structures up to 400 times in magnification in comparison to the naked eye. Okay, and this is generally what we would use in the laboratory. Of course, we're not in the lab. We're not going to be working a lot with microscopes here, but we're going to be looking at the slides, the images that we would have seen under those microscopes anyway. All right. Now, early on, we would take samples of tissues and we would actually try to look at them and had a very difficult time seeing any of the detail inside of cells. We couldn't even see the nucleus. And it so happens that the textile industry, believe it or not, and the idea of applying dyes to fabrics to give us all these different colors of our clothing actually gave some scientists some crazy ideas that we could apply these dyes, these stains, to these samples of tissues we had that they were having a very hard time seeing anything in these. And as soon as they added these dyes, these stains, which actually was borrowed from the textile industry, this greatly improved the contrast of what was in these cells, what was in these preparations. Okay? So I wanted to give you an idea of what goes into producing these slides, so that you have an idea of, where are these pictures coming from. It's out of nowhere here. So I'm going to give you a short story of a real story of someone I know. I usually start off with the students asking, okay, have you ever given a tissue or a sample of your body for examination? And a lot of times people will think of maybe biopsies. Another really standard one would be when you give a blood sample, right? A lot of us have given a sample of our blood, and then they'll be analyzed for various things. Some of these tests don't involve microscopes, but some of them do. And so the example I want to share with you, a person that I knew in grad school had married another friend of mine, actually, at the time, and they were trying to have children. And they found that they were having some difficulty, and they checked the gentleman out, and they looked at his sperm and activity of his sperm, and that seemed to check out and was fine. But then they checked out also the females' reproductive structures to see that everything was good there. And they actually found that on the uterus, and this is just an image of the uterus. I'm not gonna, this would be kind of a front-on view of the uterus down in the lower abdominal pelvic region in a female. You would find on the inside the uterus, okay, so this is basically the uterus right here. And then we'd find fallopian tubes and ovaries, and we'll talk about this in more detail later. But the point here is that they found some growths that were in a portion of the fallopian tube and the uterus for her. And so they wanted to get a better idea of what was going on there. Is this possibly the cause of their complications, her inability to conceive, to have children? And so what they wanted to do, what they did was they took a little sample here. Okay, so now keep in mind the uterus is in fact an organ. Okay, so they're taking a sample of some part of this organ. Let's keep in mind our hierarchy of complexity here. We start with atoms and molecules and compounds, chemistry stuff, right? We get up into the cell, and then when we throw a bunch of cells together, we're going to have tissues, what this whole chapter is about. But now, in order to see those tissues, what they're doing is they're taking a sampling, a little plug of three-dimensional tissue from an organ. Makes sense? An organ is a compilation of a bunch of different tissues. Okay? Now, what they need to do to actually view the cells within the tissues of this organ is they need to slice it, and that's what they do next. So here we have actually an instrument they call a microtome. Micro, so that makes sense because you're looking at small things. And tome has to do with the fact that you're taking slices. Microtome is going to take slices of a preparation made from this little plug of the organ here. Okay, so what they typically do is they take a portion of the organ, which is made of tissues. They're going to embed that in a wax of a sort. Okay, because remember, these tissues are going to be mostly made of water. So how do I slice something that's mostly made of water? It's pretty hard. So what they'll do is they'll take this and imagine a little cylindrical plug being taken out of this. So I'll just draw something really rudimentary here. So this is like a little, I always think of it, it's much smaller than a core sample of the Earth. But think of a core sample of the Earth, they go in and they take out a cylinder of the ground, okay? And then you'll see in that ground there'll be layers of rock, layers of dirt, layers of various types of soil. Well, here you're going to see exactly the same thing, except in this cylinder you're going to see maybe different thicknesses and layers of tissues, okay? So you have to imagine that here we'd have one kind of tissue perhaps. Over here we have another kind of tissue, and these would all be in layers of this sample of the organ. Okay? Now, we can't see them in a three-dimensional cylinder like this. So what we do is we embed this in a paraffin wax. Okay? So we'll use purple for paraffin. Okay? So basically they're going to embed this in a sort of wax, okay? And so here we might have a three-dimensional, and that would have the sample in it. And then what they need to do is they need to slice this. They slice it real thin. Now, here you see what they're doing as they slice it, okay? This microtome is like a mini meat slicer, okay? Think about what they have in the deli, okay? It's going to take this sample here, which is embedded in a paraffin wax. This is much bigger sample than they would have taken from the person I know, because they want to leave this organ intact to heal once they take a little sampling. But then they're going to take these slices. So here we see one slice, okay? And then they take these slices and they would lay them out on a microscope slide. Now, at first, these tissues aren't going to look anything like what you see up in the top left here. These are a nice purple, pink. Obviously, there's a tissue there. But before we started applying stains, these thin slices, you can see anything. When we put this on a microscope, a light microscope, you'd see hardly anything. But then they started to apply some of these stains that would stick to the nucleus. The nucleus is really a draw for some of these stains. And other structures as well. And then everything would light up. Okay? So these stains really gave us the ability to add contrast and see what's going on in these cells. All right. So what was the end of the story? Okay, so what they found was that there were certain tissue types that seemed to be growing abnormally, and actually it's a strange set of events. There's one hospital, their technicians and the doctors there determined that it was a really dire thing. It's not easy to determine which tissues are there, especially if which tissues are growing abnormally, right? Because the cells are going to look strange. So actually, some of the different hospitals that got these tissues, these slides, disagreed with which tissue was growing abnormally. And the good thing is, it turned out it was not a cancerous type growth, but it was actually a benign growth, and she now has children of her own. So everything worked out in the end. But this is kind of the basic way they go about making these slides. All right. So last thing I want to kind of go over with you before we really launch into each particular class of tissues would be, what are the primary tissue types? Okay. So there's 4 of them. Now, what I always ask my students when I talk about that example, I'll pose this to the entire group and I'll say, okay, do you figure that there is a muscle associated with the uterus? And everybody usually says, well, yes, right? Because there's labor contractions, right? We have muscle associated with the uterus, absolutely. So muscle. I will always ask them, okay, do you think, is there a surface to the uterus? Is there, a boundary to that organ? A surface? And absolutely, yes, there's an outside surface to the uterus, and there's actually an inside surface to the uterus as well. So there's a bunch of surfaces, right? Coverings. Epithelial tissues are always covering or lining body surfaces. Okay, so this works out. I will ask, and usually pose this to the females in the room, is there sensation associated with that organ? Can you feel that organ? Is there some kind of nervous system, in other words, built into that organ? Okay. So yes, there's muscle. There are forces that are placed on the insides of that uterus by muscle. There's nervous tissue because there's sensation and electrical signals that are sent to the brain that allow you to actually sense things there. There can be pain sensations depending on, during labor, for example, that will be shooting up to the brain that certainly will register with the female. And then there's a glue that ties all of these tissues together. And I think of that as, it's a connective tissue. It's going to kind of be the glue that holds everything together. So we've got epithelial tissues associated with throughout the body. These are going to either cover or line body surfaces. It doesn't matter if it's the uterus, or if it's the surface of our skin, or it's the inside surfaces of our mouth, or all the way through our digestive tract for that matter. We're going to have surfaces. We're going to find different types of muscle tissues that always are going to exert physical forces and cause something to move. Okay? We're going to find nervous tissue throughout the body. And we're going to find connective tissues, which are going to be like a glue that holds everything together, but they're also very diverse. They're going to support and protect and store. Some of these are actually fatty tissues that are considered connective. So the storage of energy would be part of that. All right. So what I'm going to do with you guys, I'm going to march through each one of these tissue types. Big thing now for this particular section is you will need to be able to identify by sight the various types of epithelial tissues, muscle tissues, nervous tissues, and connective tissues that I'm going to cover in the next couple of video clips, okay? I don't do that a lot in our lecture exams, but here we will have to have a visual recognition of the different types within these groups, okay? So for right now, I think that we'll leave it at that, and we'll jump into epithelial tissues next. Our next video clip, muscle and nervous tissue, and connective tissues. We're going to kind of go through these in the next, I think, 3 clips. We'll cover it. Alrighty?